Good morning. Myself Mohammed Rais PC from the Department of Electronics, Cochin University of Science and Technology, QSET, Kochi, Kerala, India. Here we are proposing a novel concept, a proof of concept, portable, non-invasive and wireless device for the preliminary diagnosis of preeclampsia. As all of you know, the hypertensive disorders of pregnancy, HPDs, are one of the main factors affecting the maternal mortality globally, and early diagnosis of such diseases can lead to a significant reduction in the maternal and fetal mortality rates, among which preeclampsia is one of the major cause of this kind of HPDs, and prevalence of hypertensive disorders of pregnancy is around 7.8% with preeclampsia conditions. So the number of preterm births reported in India is highest in the world, so among which most of them are related to this preeclampsia conditions. So it typically occurs in the third trimester, and the early diagnosis is very essential for the maternal and fetal health. The current practices in India is to identify the preeclampsia is to use the blood pressure measurement to identify the pregnant woman with a risk of preeclampsia or other kind of hypertensive disorders. So the blood and urine analysis is the only current possibility to identify such diseases. So however, the very shortage of adequate laboratory facilities in the rural villages in India is a major bottleneck in the diagnosis and measurement of the preeclamptic woman. So the proposed solution to address this problem is a device, a portable device that can measure all the relevant biophysical and biochemical parameters relevant for the preliminary screening of the woman with preeclampsia, and which can be operated by the help of a semi-skilled health workers. So the proposed device is basically a portable device, so which is wireless in nature, and it can integrate the measurement of the biophysical and biochemical parameters relevant for the screening of the condition preeclampsia. So the work proposes basically three subsystems. So first one is developing a novel strip-based electrochemical biosensors for the measurement of creatinine and uric acid, so which can be identified from the urine and blood samples. So the second part is the development of fluorescence-based biosensors for the measurement of albumin and in the present in urine samples. The third part is measuring SFLT1 or PIGF ratio, which is a novel approach using the fluorescence-based techniques. So the development of this proposed device is very multidisciplinary, and it involves a collaboration between the various research facilities. So as a primary step, we developed a prototype device that is portable and wireless, and that determines the blood pressure level, blood glucose levels using the electrochemical and fluorescence sensing, and the same protocol can be used for the measurement of other biochemical and biophysical parameters, I mean biomarkers such as creatinine, uric acid and albumin in the present in the blood and urine samples. So the methodology is the same, so which can be applied for the any kind of biomarkers, but the change will only come in the calibration plots of each of the biomarkers. So the prototype device having the key features such as it have three devices are integrated as a three subsystems. So the protocol we used is based on a glucose measurement. So the same procedure can be used for the creatinine, uric acid and albumin contents in the blood and urine samples. So what we are planning is to deploy the device in a rural villages which having sub health centers or the community health centers or public health centers, which it can be used by a semi-skilled person such as auxiliary nurse, midwife, A&M professionals or accredited social health activities such as ASHA workers to regularly monitor the various parameters of the pregnant woman during the various stages of the pregnancy. So the device integrates three different subsystems for the measurement of the relevant parameters. So first part is having a microcontroller based fully automatic system for the measurement of blood pressure. So blood pressure variations is a key indication of preeclampsia in early stages, especially in the time of third trimester. The second part is an electrochemical biosensor, so which is developed as part of the association with the Department of Chemistry of the Cochin University, along with a microcontroller based system for the automatic measurement of albumin, creatinine and uric acid from the urine samples. The third part is a fluorescent based immunoassay which is developed by the Department of Environmental Science of the Cochin University, and which is associated in circuits for the measurement of SLFLT PIGF ratio from the blood samples. So the electronics part, the electronics department developed the reader, the backend reader for the electrochemical biosensor and the fluorescence based immunoassay, and similarly the integration of the automatic blood pressure system. So the same protocol can be used for the any kind of measurement related to electrochemically or the fluorescence based methods, so which can be directly integrated with the system using the same protocol. So this is the model design, which is comprises of an embedded controller as a major backbone of the system. So the embedded controller, in the prototype we use an Arduino based system, Arduino Uno board based system. The embedded controller may be in the further level, it can be extended to Raspberry Pi and further extendable to FPGA based controllers. So the three subsystems, including the blood pressure monitoring system and the fluorescent reader, the electrochemical sensing unit, which is integrated with the embedded controller, and it will be having an output display panel to indicate the reading of the different sensors associated with the embedded controller. So the Bluetooth interface will also provide a interface with a mobile phone or a smart devices, and which can be further extended to a cloud storage. So in the proof of concept model, there will be having the three sub blocks, such as a blood pressure monitoring system, and second one is electrochemical strip sensor, the reader for the electrochemical strip, then the third part is a fluorescence strip reader. So it can be integrated with an Arduino Uno based microcontroller. It will be having a display unit and a Bluetooth Wi-Fi module for integrating. So as part of this Bluetooth integration, we use HC05 Bluetooth module, then for the which can be further extended to a mobile interface, and for which we have used some Arduino compatible Bluetooth applications. So the first part of the device is an automated blood pressure measurement system. So the method we employed in this is an oscillometric method that is commonly used in the various kinds of Omron based pressure sensor devices that is available in the market. So the basic block diagram of the system is shown. So we will be having a microcontroller, then which will be having a supported by a motor to manage the cuff pressure that is attached to the this arm cuff. So then that will be detecting the blood pressure, the cuff pressure actually detecting, and that will be properly conditioned by the instrumentation amplifiers, and then it is converted to a digital format using the ADC and given to the microcontroller. So we'll be having an LCD display to get the different reading of the pressure, which is properly calibrated with the normal blood pressure values as shown in the second figure. So the cuff pressure waveform of the oscillatory method is shown in this figure. So it will be having both the systolic and diastolic pressures that can be easily identified from the using this oscillometric method. So the mode of operation of such a device is as shown. So in the hand we will be placing this cuff and measuring the pressure, and the method is very simple. That is, the oscillometric technique is usually it is very popular in the normal market itself. So we use the same procedure for implementing this automatic blood pressure monitoring system as part of this device. So the blood pressure system will having this cuff is placed in the patient's arm, that is cuff is in the bladder is inflated with the air until it is external pressure exceeds the intra-arterial systolic pressure value and the arterial flow past to the cuff ceases. So the cuff bladder pressure is slowly released, then the pressure sensor inside the cuff detects the arterial pulsations as oscillations. As the cuff pressure declines, the oscillations increases and the amplitude is maximum, and this represents the mean arterial pressure map as shown in this figure. So the oscillation amplitude then declines until the minimum value, and the only pressure actually measures is the pressure at the point of maximum amplitude, which corresponds to the map value. So the systolic and diastolic pressure values may be calculated as the pressure at which the amplitude are fixed percent of the maximum oscillation. So this figures as shown is giving this the peak values of the systolic pressure and the mean blood pressure and the diastolic pressure value. So the next part is the electrochemical sensing, for which we have used different types of glucose strip based sensors available in the market, and the final device strip sensor we used is developed with the sensor, which is having 3 electrodes as shown in the figure. There will be a trigger electrode and a working electrode and a reference electrode. So for the experimental purpose, we just used the glucose as a biomarker, and the glucose concentration starting from 30 milligram per deciliter to 300 milligram per deciliter tested, and we have received a voltage value of 0.8 millivolt to 8.6 millivolt, so which is a proportionately increasing thing. As per the glucose level concentration increases, the voltage output also increases, so we'll getting a linear relationship between the glucose concentration and the output voltage. The same protocol is employed for the other kind of biomarkers also. So not only the voltage based analysis, we have done some impedance based test also, the concentration versus the resistance chart also produced. So in which for the 60 milligram per deciliter glucose we will getting a 75 ohms, and for 300 we are getting 165 ohms. So this also can be used as an impedance based test, also can be employed as a method for detecting the electrochemical changes. So in detail, so the electrochemical strip based detection we are using for the creatinine and uric acid detection. So as a prototype we just use glucose only, and the same procedure can be implemented for the creatinine and the uric acid analysis. Electrochemical changes in the biomarker after applying the biomarker samples induce a small current changes across electrodes. So this current signals is detected, amplified and converted to the voltage signals by a trans impedance amplifier circuit. So the voltage signal generated will be in proportion with the concentration of the applied sample. So this is the working principle of the electrochemical strip sensing. So the initial phase, as I said already, we are using the glucose solution for the reference, and a glucose solution of the non-concentration in the milligram per deciliter unit was applied to electrochemical strips, and the voltage changes are recorded, that is indicated in the table as shown. So the method is very simple, and the same procedure can be implemented for the other kind of biomarkers as well. The device is tested for random samples of the unknown concentration and verified the result using some standard glucose testing instruments. We just calibrated the value we acquired from the random samples and just verified with the instruments, glucose testing instruments available in the market. So the same experiment procedure can be repeated for other biomarkers also. So the linear relationship between the concentration and voltage values in the case of glucose is expected for the other biomarkers as well. So the impedance based testing also carried out with the samples, and the matching plot of resistance across the electrodes versus the glucose concentration also obtained, that also mentioned in the concentration versus resistance chart. So we will be using the same protocol for the other biomarkers also. So the difference will be only in the calibration plot. So the relationship between the voltage values and the corresponding concentration of the creatinine and the uric acid. Okay. In the next part we are using a fluorescence based sensing, so in which we will be having a microcontroller and an excitation unit to excite the light to the sensors. So the fluorescence based strip will be detecting the excitation, and it will be resulting a photodiode sensing approach, and the signal acquired, the light signals, and then it is converted to electrical signals, that is conditioned by a proper signal conditioning system, and then we will be having an ADC to convert into digital format, and finally it will again giving back to the microcontroller unit, it will be displaying as it is. So here we are using three kind of sensors. First method is an TCS 34725 RGB color sensor. Second one is TCS 230 color sensor, and then another last one is a laser and LDR based approach. So in both cases we will be getting positive results, and that is in proportion with the biomarker. So the design and implementation of the sensors, and which is involves the development of a system for the detection reflected or observed light passing through the sample using a photodetector, the fundamental principle of a photo detection. So the fluorescence based sensing we are using for the axial case, it is for the albumin content in the sample. That is a very novel approach to detect the preeclampsia. The part is under development with the Department of Chemistry and the Department of Environmental Sciences. So in the prototype model we are using the same glucose samples for the detection of this kind of devices. So the glucose samples will be using as a biomarker for the testing purpose, and the same thing will be implemented, the same method will be implementing for the rest of the biomarkers. So in the prototype model we implemented two fluorometer models. The first one is a laser and LDR based approach. So the LDR we use is a GL5528 sensor based detector, and the second one is a TCS 35725 RGB sensor and the TCS 230 color sensor as shown in the figures. So the glucose solution is non-concentration is then considered as a test solution, and the voltage output is in proportion with the sample concentrations recorded, and further it is analyzed. So in the first model, the sample solution is placed in a transparent glass cuvette, and the laser beam is allowed to pass through it, and the light is coming out from the cuvette is then detected directly using the LDR, and then the generated output is corresponds to the concentration of the solution. So its generally signals are then again converted into a range of 0 to 5 volts for the EC calibration. So the results we are obtaining for the direct incident laser beam, we are getting 1.66 volt. So through the cuvette we are getting the 1.60 volt. So the laser through the glass cuvette with the solution, glucose solution, we are getting a value of 0.9 to 1.20 volt. Then for 120 milligram per deciliter glucose solution, we are getting a value of 0.97. For 180 milligram per glucose deciliter glucose solution, we are getting 1.1 volt of LDR output. So this is the method we employed. So the second method contains the color sensor TCS approach and the photodiode using, so which is also getting different output. The output signals the response to the sample concentration generated as a result. So both the fluorometers giving satisfactory performance in the terms of the reflected light intensity and corresponding voltage values in the proportion with the concentration given the sample. So the final part is the NDR system, that is a microcontroller unit. The Arduino Uno board is attached, and the cuvette and the LDR system as shown in this figure is placed within this closed chamber. The three subsystems are integrated with the Arduino board through the mod switches, and it can be output displays on the mobile or tablet device connected to the Bluetooth. So in which the final stage will be having output will be each subsystem will be displayed on the mobile or tablet interface of the system using the HC05 Bluetooth module to enable the wireless connectivity. So this is a working model in which the laser beam is incidenting and the material is placed in this glass cuvette. So it's also shown here, and the LDR will be giving some output, and that will be detected by the Arduino microcontroller, and that is given to the microcontroller display unit. So similarly here we can see the TCS 320 based color sensor. So again another sample is placed in this glass cuvette, and which is also placed in a closed chamber in order to avoid that the light reflection conditions. So the entire the system will be looks like in the first figure. So the electrochemical strip reader you can see here, and this one will be detecting the signals. Corresponding signals will be amplified and properly conditioned with the help of this microcontroller unit. So the NDR system is 3D printer for the output covering, and which is again manufactured using the 3D printer available in the department. So the proof of concept device for the preliminary diagnosis of preeclampsia is developed as per this. So in conclusion, the conventional methods are available for the analyzing the biophysical and chemical parameters are very costly and which require expertise with the technical skills for operation. So there is a huge need for the portable device which can be used to diagnose the accurate clinical information of the patients for the medical purposes in the low resource settings, especially HPD such as preeclampsia, especially in the rural villages in India. So here we developed a proof of concept model as a preliminary primary prototype as a portable wireless device for the early diagnosis of HPDs. So we were able to monitor the blood pressure value, develop an electronic reader for the electrochemical strip biosensors, and a reader for the fluorescence sensing device, interface the three subsystems with an Arduino Uno based microcontroller, which will be further extended to an FPGA based very compact device and very suitable device for the purpose, which is integrating all the further biomarkers like creatinine, uric acid, albumin value in the urine and blood samples, and the display the readings in the Android mobile phone with the help of a wireless Bluetooth connectivity. And again we can use the Zigbee based modules for the Wi-Fi connectivity as well, which can be further extended to a cloud based storage for the proper data analysis. So the device will be deployed in the with the help of ASHA workers will be deploying in the rural villages, and in which will be having the public and community health centers. So the entire data will be processed, and it will be undergo the statistical analysis for the further analysis of the case of preeclampsia and moreover in HPDs in common. So we hope the final device for the early diagnosis of preeclampsia will be a great healthcare innovation, and it will definitely help the women in the rural villages. So thank you all for hearing this, sharing this valuable contributions developed by the department and the team of researchers from the Department of Electronics. So this project is basically it's a development of a portable device for the preliminary diagnosis of preeclampsia. It is supported by the Department of Science and Technology DST, Government of India, under the DST BDTD scheme. The project for the biomedical device and technology development. So we thank DST for the financial assistance and the Department of Electronics QSAT for the infrastructural and technical supports for the development of the primary prototype. The device will be completed within a span of 2 years. The entire project will be finished, and the portable device will be deployed in the rural PHCs for to support the rural community. Thank you one and all for hearing the session.